Right. We have talked about the metabolism of glucose by looking at glycolysis, gluconeogenesis, glycogenolysis, and we've looked at the anaerobic fates of pyruvate at the end of glycolysis, converted to ethanol during fermentation in yeast, or converted to lactic acid in us for the purposes under anaerobic conditions of replenishing the NAD+, which is the limiting reagent of glycolysis. Now we're going to talk about another fate of pyruvate, and this fate is mostly considered an aerobic fate, something that happens in the presence of oxygen, and that is the shuttling of the carbons of pyruvate into the Krebs cycle. And so if we look, let me get a pointer here. If we look at the Krebs cycle over here, or the citric acid cycle, we can think of it as really 8 core reactions that make up the cycle. 1, 2, 3, 4, 5, 6, 7, 8, to go back to the start. However, in order for the carbons of pyruvate to actually enter these 8 reactions of the cycle, they first have to be converted to acetyl-CoA. And so what we're going to talk about today is what I think of as reaction 0. And that reaction is catalyzed by an enzyme called pyruvate dehydrogenase, and it's really the pyruvate dehydrogenase complex that is catalyzing this reaction. Now, if we look at reaction 0 on this slide here, we can see we're starting with pyruvate at the start. We're converting it to acetyl-CoA, which is a 2-carbon acetyl group linked to a vitamin, really, a cofactor called coenzyme A. We'll look at the structure of that in class. Now, what are the other products of this reaction? Well, carbon dioxide is where that 3rd carbon from pyruvate ends up. And again, we make a reduced cofactor called NADH. Now, the reactions of the citric acid cycle and reaction 0 are going to be happening in the mitochondria, and we'll look at mitochondrial structure in a few topics from now. And so we're going to think of this reduced cofactor NADH a little differently than the cytoplasmic NADH we made in glycolysis. Okay, but the net reaction of reaction 0 is that pyruvate gets converted to acetyl-CoA, producing carbon dioxide, NADH, and we need some of that vitamin, that cofactor CoA, to be input into this reaction. Now, this reaction 0 is incredibly complicated, incredibly complex, and it is catalyzed by this multi-subunit enzyme complex, the pyruvate dehydrogenase complex. And it is an impressive machine. There are actually 3 enzymatic components in pyruvate dehydrogenase complex, E1, E2, E3. You can think of these almost as separate enzymes, but they're bound together into the PDC, the pyruvate dehydrogenase complex, and work together to facilitate this overall reaction 0. Now, why do we have such a big, crazy complex with all these enzymes sort of stuck together? Well, the thought is that during evolution that it was favorable to have these separate enzymes come together, and you can think of it as almost like putting together production into one factory, right? As enzyme 1 does its job, the products can be immediately placed at enzyme 2, which the products can be immediately put onto enzyme 3. And so we think of the formation of this multi-subunit complex as allowing for the products of one enzymatic action to be sort of handed directly to the next enzyme and to the next enzyme. And that's a more efficient process. And this is a really, really, really important process because we think of reaction 0, or the pyruvate dehydrogenase complex, as really the gatekeeper to the Krebs cycle. Are the carbons of pyruvate going to be put into the Krebs cycle or not? This is the complex which decides it. and therefore it's important that it's really efficient, and we'll find that it's really highly regulated also. Okay, so good grief, look at this. This is a big mess. We're going to have to piece apart the different enzymatic activities of the PDC and try to get it straight in our head. We can look at this, these three enzymes, and really think of this reaction occurring in 5 different steps. Right? Enzyme 1 is decarboxylating, right, our pyruvate, getting rid of the carbon dioxide and tying this 2-carbon piece to a cofactor called TPP. Right? TPP holds on to that cofactor, and in that process, our 2-carbon piece becomes reduced. Right? We have a carbonyl group here on this pyruvate, becomes an alcohol group. That's a reduction reaction. The TPP just holds onto the 2-carbon piece temporarily, and then in the second step, we'll transfer those 2 carbons over to a second vitamin cofactor called the lipoamide. This looks like a disulfide bridge here, and since we remember that, this is the oxidized form of it. And so as we pass the 2-carbon piece from TPP to the lipoamide becomes reduced. Right? This looks like a reduced cysteine, a broken disulfide bond. And as that lipoamide becomes reduced, we can reoxidize this alcohol group on our 2-carbon piece back to a carbonyl. Okay. So we've passed off the TPP from enzyme 1 over to the lipoamide on enzyme 2. And finally, well, not finally, but really the last part of the real reaction is that these 2 carbons that originated from pyruvate are going to get passed on to coenzyme A. So coenzyme A will take those 2 carbons and form acetyl-CoA, and this is what is put into the Krebs cycle. Now, the rest of this reaction, right, steps 4 and 5 here, are just really to help regenerate the enzyme. As our reduced lipoamide has passed on the 2 carbons to acetyl-CoA, it stays in this reduced form, and we have to reoxidize it in order for it to pick up another 2-carbon piece from E1. So how are we going to reoxidize this, lipoamide? Well, if we want to oxidize it, we can reduce other things. And so what we're going to reduce is a cofactor called the FAD cofactor. We'll reduce it from FAD to FADH2. Right? FADH2 is a reduced cofactor, kind of like NADH. And in this case, in the pyruvate dehydrogenase complex, the holding of electrons by the FAD is just transient. It occurs just for a short amount of time, because as soon as FAD grabs those electrons to form FADH2, its reduced form, it then passes on those electrons to our favorite reduced cofactor, NAD+, forming NADH as a final product. So if I try to outline what the main products are of this reaction, let's do it in green here. We can see carbon dioxide is a product of this reaction. Right? The other 2 carbons from pyruvate sort of get passed from E1 to E2 to acetyl-CoA, which is a product, final product of reaction 0. And then, in order to restore the lipoamide, we pass electrons to FAD and then finally to NADH. So this NADH, again, mitochondrially formed, is another product of this reaction. Okay. Let's look at this with a video and see if we can maybe catch this in our head a little better. Here is our pyruvate dehydrogenase complex, which with its 3 enzymatic activities. Pyruvate passes its 2 electrons onto E1, the TPP. TPP passes those 2 electrons onto our lipoamide. The lipoamide passes those carbons onto our acetyl-CoA, And then this lipoamide is reoxidized to form the cyclic form by reducing FAD to FADH2, which then passes those electrons from NAD to NADH. Okay, let's see if we can put this together in pieces. So here's the real mechanism of this, which you're not responsible for, but it shows sort of exactly what's happening and how these cofactors are really, really important chemical functional groups that are present in these enzymes. Okay, so here's E1, and this is the TPP. This is a thiamine vitamin. A lot of vitamins that you take have thiamine in them, and this is the functional part of the TPP, and it's what's going to accept the 2 carbons from pyruvate. Okay. So it forms a carbanion, and that carbanion is going to attack the carbonyl group of the center of pyruvate. Carbon dioxide is going to leave as a leaving group. We've got these 2 carbons now attached to TPP, and that carbonyl becomes reduced to form this active acetaldehyde group. Now, remember the next step is we're going to pass these 2 carbons onto the lipoamide. So let's do that with a little bit of help from a nucleophile, which again generates another carbanion. This is a great Lewis base. It's going to be able to attack the lipoamide, which starts off in the oxidized form. As those 2 carbons are passed over to the oxidized lipoamide, the lipoamide becomes reduced, breaking that disulfide bond and oxidizing our 2 carbons back to the acetyl. Lipoamide is going to pass those carbons onto our coenzyme A to form our acetyl-CoA, which goes on to the Krebs cycle. And now the rest of this mechanism is trying to restore the lipoamide back to its oxidized form. First, by passing electrons onto FAD to reduce it to FADH2, reoxidizing our lipoamide, and then FADH2 is going to pass those electrons onto NAD plus to form NADH, which we can think of as another output in our accounting sheet. We're going to hold on to these electrons and hopefully use them later in metabolism. Okay, the final thing I want to show you is just how complicated this pyruvate dehydrogenase complex is. It's a monster machine, okay, with 3 enzymatic components, multiple subunits of each one of those enzymes, and there's a total of 60 subunits in this thing. Now, the other impressive characteristic is that the molecular weight of it is 4,600 kilodaltons. That's 4,600,000 grams per mole. And so there must have been a real evolutionary drive to build and assemble these enzymes together into this huge machine that can accept reagents and pass products on to the next player in this factory. Okay. You can actually see this thing under a microscope, which is astounding to think about. This is an electron microscope picture of the PDC complex, and it looks kind of like a cube with these enzymatic components sort of decorated all on the outside of this cube. But it's such a huge structure, you can actually see the thing under a microscope.